Good. Okay, well, thanks for having me here. What I want to do today is I want to tell you a little about the research project we've been involved with monitoring the horizontal levee. And I guess it's a little bit of a story. And the story I'm going to tell you is the way I inserted myself into this project early on and brought in some research on the water quality aspects and the results that we've been getting so far and how it's changed the way we think about the project from just being a demonstration project to being something that includes research that will inform decision making about future horizontal levees that might be built in the Bay Area and elsewhere. And so I want to start by acknowledging my students, Aiden and Angela. Maybe the staff here has seen them out and about on the levees, and we're about to go into the summer season, and you'll see some more undergraduates, because we normally take on a series of undergraduate trainees over the summer who come as part of our NSF Engineering Research Center and join the research project. So you'll see even more activity in the summer, and those of you who are around on Tuesday will see about 80 or 90 people from our Engineering Research Center, because we're having our annual meeting, and this year the field trip before the annual meeting is to come here in the morning on Tuesday. So you'll see a bunch of people, but we'll have that under control. So this is how I first got involved in this project. I went to a meeting of BACWA, and you all know what BACWA is, the Bay Area Clean Water Agencies, and saw this fellow here, Jeremy Lowe, presenting on the idea of the horizontal levees just when it was a research proposal to the IRWIMP program. And he was talking about the way in which the horizontal levee would protect coastal environments from flooding and also would provide habitat. And there was kind of like an, oh, by the way, it's going to improve water quality. And after seeing him present this, I said, well, tell me more about the water quality. What are you doing? What are the research objectives? You know, where is this all going? And he said, well, I'm a geomorphologist. My colleagues are plant biologists and wetland ecologists. We're trying to figure out exactly what we want to do with the water quality part. And I said, oh, I volunteer to join the team. And so I kind of inserted myself into the team and the UC Berkeley team, not just myself, but Professor Alex Horn, who's an emeritus professor at UC Berkeley. And we were involved in the original conception and design of taking it from the proposal stage to something that could actually be built. And of course, you're all familiar with the location just outside here. The circle shows what the site looked like before the horizontal levee was built. But this is kind of the cartoon or the schematic of the system that I think we're all familiar with, where we have this wet weather basin, which also functions as a surface flow wetland, and then the horizontal levee. And the attributes of it that were included to improve water quality are as follows. First, this idea of the nitrification trench, because the wastewater treatment plant is producing ammonia in the effluent, and ammonia is toxic to mosquito fish, and so we felt if we were going to have a surface flow wetland, we wanted to have water that would work for growing gambusia or mosquito fish, so we could eat the mosquito larva, or the fish would eat the mosquito larva, so we wouldn't have a vector problem. And so the nitrification trench took care of that. And then those mixing zones, or refugia, are areas that the mosquito fish can go into and in times of high water temperature or low dissolved oxygen, and it serves as a refuge for them from predators as well, from birds and things like that. And the second aspect of the project, which we played a role in helping to build in, was this idea of, instead of just studying one horizontal slope, to break it up into these 12 independent cells so we could study the effects of different kind of fill material and different kinds of flow patterns. And if you go out there and you look closely, you'll see that there are actually 12 little environments in there, and they each have a slightly different treatment conditions. Some of them have willow trees, that's pretty obvious. The other ones are a mixture of different types of plants and different kind of sediment substrate materials, and that is relevant because if we want to build this thing at scale, we'd like to know what kind of sediments we should use and what kind of plants we should use. So it's a rigorous research program because these treatments are done in triplicate, so we can see how much of the variability is due to the variability of the location or the local biology, and we can test different hypotheses about the wetland system. And so the research objectives, or the monitoring plan, is really testing some real research objectives about the wetland. The first and foremost is the effect of the wetland on nutrients. And the main thing that we hypothesized here was that the wetland subsurface environment, where the water's flowing, had the chance to remove nitrate by the process of denitrification. And denitrification is the process where microbes, instead of breathing oxygen, they use up all the oxygen and they breathe in nitrate. And they use that as a terminal electron acceptor. Maybe you remember that from high school biology. And they breathe out N2, nitrogen gas, the gas that surrounds us and is mixed with oxygen in the room here. And that process takes a nutrient that's a contaminant that could cause algal growth or excessive algal growth in the bay and turns it into this benign form of nitrogen that we all breathe in the air. So denitrification is something that we hypothesized would be important because under the subsurface, in the subsurface of the wetland, there should be no oxygen because the microbes use up all the oxygen. The other thing we wanted to think about, though, in studying this as a research project is the possibility that some of the nutrients would be taken up as plants. Obviously, you're growing plants, you're passing water full of nutrients through. Maybe you're not seeing any removal of the nutrients at all. Maybe it's just accumulating in the plants the way it does when you put nitrogen and phosphorus in your garden. And with the phosphate, there was this other aspect we wanted to study, which was the fact that phosphate tends to stick to the surfaces of iron oxides, and iron oxides are very common in soil and sediment, so that was something to pay attention to. Mostly we're focused on nitrate, and the reason we're focused on nitrate or nitrogen removal is at San Francisco Bay, the limiting nutrient is nitrogen, and the San Francisco Regional Water Quality Control Board is very concerned about this and is contemplating putting discharge limits on wastewater treatment plants for nutrients, and that could turn out to be like $1 billion problem for the Bay Area treatment plants because it would require retrofitting with nitrogen removal capacity. So the subsurface wetland or the horizontal levee has the potential to obviate the need to actually upgrade a treatment plant. It actually would take care of something and you wouldn't have to do an upgrade to a treatment plant. The second thing that we were interested in is the potential for the subsurface to remove what we refer to as trace organic contaminants, and maybe you've heard people talk about the fact that wastewater effluent sometimes contains residues of pharmaceuticals. It also contains things like urban use pesticides, like when people put Frontline, which is fipronil, on their dog and cat, and then well, you don't wash a cat in a bathtub. I've heard that's a bad idea. I'm a dog owner, and I know people wash their dogs now and then, and that fipronil goes down the drain, and the city of Palo Alto has seen pretty high levels of fipronil in their wastewater effluent. So it's not just pharmaceuticals, it's personal care products we use in the home, and it's pesticides and things like that. And we've been studying the removal of pharmaceuticals in wetlands for a number of years, and in the subsurface wetland we had 3 different hypotheses. One is that they could be removed by degradation by bacteria. They're bacteria living under the surface. They might metabolize these things. They might stick to sediments. They might attach to the surface of sediments, and they might be taken up by plants because plants are sipping that water. If the pharmaceuticals are dissolved in that water, it can go up and into the plants. And finally, in terms of the research, we are very interested in understanding the operation and maintenance of the system, because if we're going to scale this out and build a bigger horizontal levee, we need to know how to build it so we can minimize the cost of maintenance. And so this idea of the rhizosphere development, the rhizosphere is just a fancy word to say the roots underground. And one of the ways in which this system works is that those roots of the plants that grow, as the roots die, they're a source of organic carbon that feeds the microbes. So we're very interested in how that rhizosphere develops. And then finally, the hydraulics of the system. I'll tell you a little bit about what we've seen, but certainly, especially the willow plants, they have roots, maybe those are the same roots that get in and cause mischief in sewer lines. Those willow roots can cause mischief in our water distribution system, and we're studying that to try to understand the maintenance protocols. So any good research project needs a conceptual model, and this is our conceptual model of what happens. The water goes in at the top here, and you could think about it, it's wastewater effluent going in at the top, and most of it's flowing through the subsurface, because when we built the horizontal levee, we had a layer of coarse sand and gravel in the bottom here, and the plants themselves are growing in a clay loam layer, and there's very little water permeability. So most of the water is flowing in this subsurface layer, and the bottom here is a clay layer, which means nothing really flows underneath that. And then when we built it, we put wood chips in, and the wood chips are rich in organic carbon, as you can imagine from wood chips. chips. And the microbes grow on the surface and form a biofilm, and that biofilm is where this breathing of nitrate happens, or denitrification. So the wood chips are there to jumpstart this process, but over the long term, the roots of the plants are going to contribute the organic carbon that's going to fuel this denitrification process. And of course, after the water passes through the system, it gets returned to the headworks of the treatment plant. So, you know, any research project is always handicapped by reality. When you actually start studying the project, you learn some things that maybe you hadn't been thinking clearly enough before. And this was one of the things that initially puzzled us and turned out to be a source of knowledge for us. We started out by measuring chloride. So here are the 12 cells, and if you go out there, you'll see there are these sampling ports at 15, 30, and 45 meters through the system, where you can collect a sample through a gravel trench that kind of looks like a French drain. And we sampled chloride, and chloride should not undergo any chemistry. It's an inert tracer of the water. And what we saw is that the chloride concentrations went up by about a factor of 3 as the water passed through the wetland. And if you think about that, that kind of makes sense, because the wetland's full of plants. The plants are sipping water, and that water is going out into the atmosphere through the process of evapotranspiration, and it leaves behind the salts in the water, because the plants sip the water and leave the salts behind, so the salt concentration builds up. This is kind of like if you've heard people talk about salinization of agricultural soils. It's the same phenomena. If the plants are using the water and the water's evaporating, the salts build up. So the salts built up, and then they came back down at the end. And what we realized what was going on there was that the sampling point at the very end, and if you go down there and have a look at it, you'll see it collects all the water, not only the water that's been flowing in the subsurface, but the water that's been flowing over the surface. And what we learned in our first year of monitoring was that for some of the cells, a considerable amount of the water was actually traveling as a thin sheet of water across the surface of the horizontal levee. And then some of the water was traveling in the subsurface like it should. And what we learned was that that water that flows along the surface does not lose its nitrate, because it never goes into an oxygen-free environment where the microbes can denitrify it. So it just flows over the surface, and we get poor nitrogen removal. So one of the things we learned in the first year is that if we want good removal of nitrate, we have to keep absolutely all the water under the surface and avoid that so-called overland flow. And so if you just think about the difference between the first sampling site and the last sampling site, you can think about this ratio of the chloride going out to the chloride going in, and you can see that it's about 2 throughout most of the year. We've continued to monitor this, but I'm showing you the 2017 data, and you can see that when it's cold in the springtime, it's a little lower, and then in February and March, it went down a little bit too, because the plants become less active in the wintertime. But basically, about half the water we put into the wetland just goes out into the atmosphere. The other half gets discharged. And that is kind of interesting to us in terms of future designs, because it tells us about how much water will actually be discharged to the bay. So what you can see here is some data on the nitrate remaining. So this is kind of like if there's no removal, it would be up here at 1, and if it was all removed, it would be down at 0, as a function of the 4 different kinds of treatments. And what we saw in this is that we had pretty good removal last year between April and May. We were seeing greater than 90% removal, and then after June, the removal went down a little bit. And what happened there is we had turned up the flow a little bit, because the plants were using more water, so we turned up the overall flow, and a larger fraction of the water passed through the wetlands through this overland flow. And so we can use the chloride data I was showing you before and categorize these different wetlands in terms of the fraction of overland flow. And what we see is if we group the wetlands that have no overland flow together, we're getting essentially 100% removal of nitrate. And when we look at the wetlands, for example, the first 3 when you walk in, which we call the wet meadow, when we look at those wetlands, which were designed to have like a stream trickling down the center, we see only about 40% of the nitrate being removed, because so much of the flow is along the surface, and the other ones fall somewhere in between, depending upon whether the roots of the willows clogged the inlet structure and forced the flow to go overland, or the flow was just flowing overland because it was a fine-grained sediment material material, and so there wasn't as much ability of the water to flow in the subsurface. And I could show you the data, the monitoring data on the nitrate, now that we've got the water all flowing in the subsurface, but it would be incredibly boring, because now that we have all the water flowing in the subsurface, the nitrate disappears within that first 15 meters, the first third of the cell. So we have a bunch of non-detects for nitrate now that we've got it figured out. The second thing that we've been doing to try to understand this issue of whether the nutrients are being taken up by the plants or whether they're actually being removed by these chemical processes is to try to understand the growth of the plants. That is, in the first year, if you went out and looked at the wetland, it was a bunch of twigs that we put in the ground. And then by the end of the first fall, they were little tiny bushes. But if you go out there now, you see that they look kind of like mature plants. And our hypothesis is that now we're in the third year or so of the wetland, we probably are reaching a steady state where the plants have reached their maximum amount of growth. And the way in which we're verifying this is twofold. One is we do the old school quadrat biomass harvesting, which means you either find a class of students or you offer a bunch of graduate students free lunch and maybe beer afterwards, and you go out and you look at these 1 meter plots and you shave off all of the plants and you weigh them and you see how much biomass is there. The second thing that we're doing is we're actually doing drone mapping, where we're flying a drone with an infrared detector, and we can correlate that infrared radiation to the amount of plant growth and map the amount of plant growth over time. And that's kind of the direction we're taking now, because this technique is so labor intensive. The other way in which we're trying to gain insight into what's happening here is by using stable isotopes of nitrogen. So if you could remember from high school chemistry or physics, all of the elements in the periodic table have multiple isotopes. There are isotope mass spectrometers that can tell the difference between the different isotopes, and those isotopes behave preferentially in biological systems. So to make a long story short, Aidan here, who's doing a lot of isotope work in his thesis, was sampling the willow cells last summer and looking at the stable isotopes of nitrogen. And one of the cells, cell H, looked the nitrogen in the willow plant leaves all looked like the wastewater signature. And we found that that particular cell, the roots of the willow trees had clogged the inlet, so most of the water was flowing over the surface, and there was always ample amount of nitrate for the willows to sip. This cell here, cell L, was functioning the way it should, and so the willows at the very front end showed the isotope signal of the wastewater, and the willows starting about a third of the way into the wetland showed the native background soil nitrogen signal. That is, they were getting their nitrogen from the nitrogen that was in the soil from the wood chips and that was originally there. And then this other one here, J, which had a clogged inlet and which was actually quite dry last summer, it all looked like the soil flow because there really was no wastewater nitrogen coming in. So this very powerful tool of stable isotopes of nitrogen is allowing us to understand exactly where the nitrogen's coming from and what part of the wetland it gets used in. Looking at the trace organics for a minute, we've sampled a suite. Here I'm showing you 7 different pharmaceutical compounds. Some of them are antibiotics like sulfamethoxazole, atenolol and metoprolol are beta blockers, heart medications, and then acyclovir and tenofovir are antivirals, and carbamazepine is an anti-epileptic drug. All of these things go away to some degree in the surface flow wetland, in the surface overflow with the cattails and the bulrush, but some of them not so well. So for example, sulfamethoxazole and carbamazepine and tenofovir, these were barely removed. There was only about 10 or 20% removal of them in the surface flow wetland, but when they hit the subsurface wetland, they were all removed. And this is pretty remarkable to us. We haven't really seen much data like this before. They're not removed in the sewage treatment plant where they see lots of microbes. They're not removed in the surface flow wetland. For some reason, they seem to be getting removed very quickly when they hit the horizontal levee, which is good news for us because the water coming out of the levee is quite clean with respect to pharmaceuticals. And what we saw is that when we looked at that overland flow issue, we saw a very clear correlation. If we could keep the water underground, we removed the pharmaceuticals. When we had flow across the surface, we removed less of the pharmaceuticals. So I promised 10 minutes. I've probably already gone over my 10 minutes because I'm totally enthusiastic about this stuff. Let me kind of tell you about the next steps and where we're going. This summer, we're very interested in the nutrients, and we're looking at the effect of temperature. So when we go over a seasonal cycle, we expect in the winter less removal and more removal in the summer. We have some preliminary data showing that as we transition from March through May, the removal improved, and that's because we're using pore water samplers and we're getting a higher spatial resolution. We're also interested in the initial concentration of nitrate. So we reverse the flow of the water going into the wetlands to try to raise the nitrate concentration, because we wanted to understand if the nitrate concentrations increase over time as people get better at conserving water. Will the wetlands be overloaded with nitrate, or will they continue to remove it? We're also interested in informing other types of wetland restoration projects where there might be higher nitrate levels. We're looking at this issue of plant maturation on nitrogen removal more seriously, so we'll be back looking at stable isotopes this summer. We're sampling pore water, as I mentioned. We're not just sampling at those 3 different sampling locations, but we go in with something called a pore water peeper, and we can look at different locations and at different depths. And then I didn't really have time to talk about it today because phosphorus is not as important in the San Francisco Bay, but we have ideas about how the subsurface wetland could be used to remove phosphorus. Again, not as important in San Francisco Bay, but might be important in other parts of the world. With respect to the trace organics, we're trying to confirm that the disappearance we see is really disappearance through microbial degradation. So we're looking at the transformation products. So we can measure the compounds as they break down and look at their daughter products. We're looking at the absorption of onto sediments and the possibility of plant uptake. So we're taking plant tissues back to the lab and we're digesting them in methanol, and then we're measuring the pharmaceuticals. And then we're trying to understand the rhizosphere microbes and understand which bacteria do this. And I have a colleague from Colorado School of Mines named Josh Sharp who's worked with me on other wetland projects, and his graduate student has started looking at some of the microbial samples. And then finally, we're very interested in the issues of operation and maintenance. So we installed these flow meters when we started the project, and we've all been struggling a little to keep them up and running, but we're really trying to get the flow meters to work well this summer so we can avoid that overland flow issue. And we've been having meetings and talking about how best to control the willows, because if this project gets built out at scale in the future, we want to be able to give people directions on where and when and how to maintain willow plants. And then in terms of future issues, kind of beyond these next 2 years, there are a couple of things we've been talking about and thinking about. One is the next horizontal levee. So our IRWIMP project had funding in it to think about where else in the Bay Area people might build horizontal levees. And so the team that's been doing that has been identifying sites and started talking to some of these other utility districts. But we're thinking about how one would reduce the construction costs. For example, bringing in the sediments was quite expensive. You might even remember that period when the dump trucks were driving up and down Grand Avenue bringing all this fill in. If we can use bay sediment or we can regrade areas, that might work better for us. And what we've learned so far is that most of the treatment happens in that first 15 meters. So we might be able to only use that expensive fill material at the very beginning until the water quality improves, and then use whatever we could find, whether it's bay sand or dredge spoils or something like that. We were thinking about how we would design those for low cost and low maintenance, and also attractive habitat. And then we're starting to think that in the future it'd be interesting to suss out this phosphorus issue, and also to think about waterborne pathogens. Again, not too many people swimming in the beach out here, but if horizontal levees are going to be used in other places, it would be very convenient if they also removed viruses from the water through those natural processes. And then the second issue that is very interesting to me, and we've been having some discussions with the San Francisco Estuary Partnership, is whether the horizontal levees could be used for treating reverse osmosis concentrate. Because the city of San Jose is proposing a potable water reuse project, and they produce RO concentrate, which is basically wastewater effluent, but 6 times the strength of what you see here. And if that could, and right now it's turning out to be kind of the biggest challenge in that project is how to remove the nutrients and organic contaminants from the RO concentrate. We have a project with the Santa Clara Valley Water District looking at something we call an open water wetland for removing those contaminants, but we think that the subsurface wetland could do the similar kinds of things. So we're thinking, wouldn't it be interesting if sometime in the future we could repurpose one of those wetland cells or a portion of one of those cells to see if it could treat the RO concentrate, because the plant species we have could live at that higher salinity, and we're now showing that the wetlands can work at that higher nitrate concentration, so it would be a very interesting experiment for us. I think that's also relevant because over time, all wastewater is going to become stronger. We're going to have people implementing conservation, and so the strength of wastewater produced indoors is going to go up, and so we're going to have higher concentrations of everything, which looks a little like reverse osmosis concentrate. So in summary, so far what we've learned is that the horizontal levee is very effective at removing nitrate from the subsurface, provided you can avoid that overland flow. And the initial data suggests that the system is somewhat overbuilt. That is, maybe we don't need such a long distance of that subsurface porous media, and that might mean that the next one we build will not require as much of this expensive gravel and sand fill. We've learned that it's capable of moving trace organic compounds, which everyone had thought at one time were recalcitrant, were difficult to degrade. And so that's kind of an interesting mystery that we're hoping to dig into. But the mechanisms of it obviously require some additional research. And then finally, in terms of operation and maintenance, the flow control has to be carefully modulated, and over the course of a year, the amount of evapotranspiration goes up and down, so we have to learn how to track the amount of water we're putting into the system with the amount of flow. The flow meters are quite useful, and hopefully optimization of this system will mean that they'll cost less money to build in the future, because if we want to build these down at the next mile south of the plant here, or over in Palo Alto with the South Bay Salt Pond project, we want to make it as cost-effective as possible. So I'll end there with the names of the team, and you can see all the people who have been involved, especially the people here at the Sanitary District who've been involved in the project, but also Carlos at ESA Associates, and Peter and the team from Save the Bay. It's been a real nice partnership among all of us. In the case of the UC Berkeley team, our work has been funded by the National Science Foundation. Aidan was supported on an NSF Graduate Research Fellowship, and more of the support comes from the NSF Engineering Research Center, Renew It, which I'm part of. And of course, Oro Loma Sanitary District, which you can see here. So we've had support from you guys all along. How did I lose that? It was here on this one. I don't know. It was on the previous slide. But of course, the Sanitary District here has been instrumental to helping us with some of the construction and some of the meters we needed and some of the support for the students. So with that, I probably lost the 10-minute bet, right? But I hope I didn't lose your attention. Okay, good. Thank you. Thank you very much.